Hello everyone, welcome back to another lecture from Shomu's Biology. Today we are going to talk about tryptophan operon and tryptophan operon attenuation. This topic is already made simplified multiple times. I have made videos regarding tryptophan operon I think 3 times earlier. Now I am making this video another time almost 10 years later again about the tryptophan operon and tryptophan operon attenuation in the same video. So I want you to give me 20 minutes of your time and I'll make sure that you'll never forget the tryptophan operon nor the tryptophan operon attenuation. So let's begin to talk about TRP operon and TRP operon attenuation. This video is for everyone who want to understand tryptophan operon from the basics, be it graduates or master students, +2 students, or anyone who is enthusiastic to learn about tryptophan operon. So starting with the idea of, we know that the operon concept is the gene regulation concept in bacteria, in prokaryotes, where more than one structural genes under the regulation of one promoter operator pair. And there is no difference that the tryptophan operon also is regulated in that fashion. So in prokaryotes, what we get as a tryptophan structure or the operon structure, what we are going to see is that these are the structural genes that we have, and in the structural gene element, let me draw it as a box, and we'll say that E, D, C, B, A. These are all structural genes, A, B, C, D, E, but in the inverse order. These are structural genes, and the promoter operator pair is present in here. This is the promoter region and the operator is somewhere inside the promoter. So this is the promoter operator pair who regulates the set of E, D, C, B, A, all the structural genes at once. But right after promoter operator pair, there is a sequence known as leader sequence, and attenuator is also present in this leader sequence. What is this leader sequence? We are going to understand that in a moment. This leader sequence is not common. This is absent in lac operon, but it is present in tryptophan operon. And further upstream, what we have regulators, the tryptophan repressor and the promoter for tryptophan repressor. Okay. So this is pretty much the simplified drawing of tryptophan operon structural section of the DNA, the structural prokaryotic DNA that we can clearly see. Now we'll understand the basics of tryptophan operon first, then we'll talk about the attenuation of tryptophan operon and why attenuation is important. Now imagine one simple thing. You imagine yourself as a bacteria. Tryptophan is an important amino acid. So you need tryptophan for certain activities in your body, in your cell. So you know XYZ, whatever function it plays, but you require tryptophan for regular maintaining a cellular activity in your body. So tryptophan is needed, and to produce tryptophan you depend on yourself to produce tryptophan. You don't need to eat other foods. You can produce tryptophan on your own. To produce tryptophan on your own, you need to transcribe this E, D, C, B, A, and the product of all this can lead to the formation of tryptophan. This is how tryptophan amino acid can be produced in your body. You don't have any issues with that. Now you want to produce tryptophan, it is necessary, but when will you produce tryptophan? When there is no tryptophan present in the cell or in your environment, then only you will produce tryptophan on your own. If you get readily available tryptophan all around you, you are not going to produce tryptophan. To understand operon is to apply your common sense. If tryptophan is already present in the environment, if it is present in the environment, then there is no point of this operon, no point of this tryptophan production, because it is already there. But if the tryptophan is absent in the environment, then it makes sense to turn on the operon to produce more tryptophan so that you can utilize them, right? That's how simple it is. So now what we are going to see is that at this particular moment, what happened in this operon is, let's imagine that there is no, there is tryptophan present, okay, and there is no tryptophan present. So if there is no tryptophan present, if the tryptophan is absent, then what happens is that this repressor promoter, from promoter the repressor is made, the repressor protein is made, but this repressor protein cannot function on its own, it cannot function on its own, means the repressor although is present, but repressor cannot bind to the operator region, thus the promoter is free, thus the polymerase, RNA polymerase can bind and can transcribe the EDCBA, all the structural genes into mRNA, and the proteins can be made. Okay, when there is no tryptophan, remember, if there is no tryptophan in the environment, then this thing is happening, then the transcription is on, the operon is turned on. Okay, because although the repressor is made, but the repressor is not strong enough to bind to the operator region. This repressor will be functional only when tryptophan binds to it. When tryptophan binds to this repressor, then the repressor becomes active, right? And when the repressor becomes active, then it can bind to the promoter operator pair. And when it happens, when there is the presence of tryptophan in the environment. When the tryptophan is present, this tryptophan is going to bind to this repressor and the repressor become active, and this repressor is going to, again let me draw here, the repressor is going to bind to this promoter operator region. So let's imagine that this is the repressor binds here. How? Because the tryptophan is associated to the repressor, making it active form, and this active form of repressor binds to the promoter operator pair, and that will prevent the transcription event, no transcription event. So the operon will be turned off. The operon will be turned off. These are the two situations. Situation number 1, no tryptophan, operon is turned on. Situation number 2, tryptophan, operon is turned off. And in this particular example of tryptophan operon, we can clearly see that the repressor by default is inactive. Okay, when the tryptophan associated with the repressor, then it becomes active. Okay, so the tryptophan molecule here act as a co-repressor, right? Co means they function jointly, the tryptophan as well as the repressor function jointly to inhibit the transcription of the structural genes of tryptophan operon. So they act as a co-repressor. So tryptophan here, let me write, act as a co-repressor. If you recall lac operon, you'll understand in lac operon the lactose act as an inducer to that operon, but in tryptophan operon, tryptophan act as a co-repressor to that operon. Okay, so this is very simple. This idea is very simple. With the basics you will understand this. Now comes the complicated part. What most of the students ask me, they don't understand the process of attenuation. Now let's go and talk about the attenuation. So here, before understanding attenuation, you need to know one simple concept. The concept is here, why attenuation is required? Because obviously this tryptophan can auto-regulate its production. If tryptophan is already present, it shuts off the production. If tryptophan is absent, it influences the production. So it's a kind of a self-regulation, pretty well sorted. So why do you need attenuation? Sometimes what happens in a situation when, because when they produce this EDCBA, all the structural gene products, the proteins, this is an anabolic pathway. We are producing something from scratch, and in any anabolic pathway where we are producing something, it consumes a lot of energy, as ATP. So if somehow in our cell or in the environment tryptophan is present, still this operon is turned on. Try to understand, if the tryptophan is present, still the operon is turned on, that makes no sense for the cell to invest extra ATP to produce more and more tryptophan, where tryptophan is already present in the environment. And this might happen, and this really happens. There are situations in the cell when although the tryptophan is present, but still it failed to shut down the transcription of the structural genes. Somehow this polymerase, RNA polymerase, can still bind to the promoter and can initiate the transcription process. Somehow the tryptophan is not well working in shutting off the tryptophan operon. There might be the problem due to the tryptophan's association with the repressor. The repressor may not functioning properly because tryptophan itself will not directly interact to the promoter operator. The repressor binds to the operator. So the repressor is faulty, or maybe the tryptophan repressor binding is faulty due to some reason. Even if the presence of tryptophan is there, still the transcription is going on, still they are making all the mRNA for the structural genes, they are making proteins for the from the structural genes. That is an alarming condition where the cell will lose a lot of energy as a form of ATP, because cells requires ATP for many other vital work in the cell. So in order to prevent this, they put attenuation in place. So attenuation is a secondary regulation or secondary control right after the operon regulation. So attenuation is a secondary control. Primarily the operon regulation is done by tryptophan. If it fails somehow, then we have a second option of attenuating and terminating the process of tryptophan operon when the tryptophan is adequately present in the environment. Keep this in your mind. This is where the importance of attenuation comes in. Although there are other operon concepts out there where it completely depends on attenuation for the regulation. An example of such kind of operon, if you want to remember, is histidine operon. If you know histidine operon, histidine synthesis again is another process of histidine synthesis, another amino acid synthesis operon in bacteria. We see histidine operon entirely depend on this attenuation. Now I will move to the next slide and I will explain what attenuation really is. Okay. And to understand attenuation, one more thing that I want you to know is this particular structure. Again, I will refer you to the structure of the structural genes out there. So these are the structural genes Z. We will write E, D, C, B, and A. All the structural genes are in place, and here we have the leader sequence. So we have this structural genes out there and a leader sequence. So the most important part here is this leader sequence. This is what we are going to put our brains to, right? So to understand leader sequence and why it is so important. And before understanding this, we need to understand another concept. In prokaryotes, the transcription or RNA production from DNA, that particular part is coupled with the protein synthesis. So we call it transcription translation coupling. Transcription translation coupling is an event that we can clearly see in case of prokaryotes, which is not present in case of eukaryotes, remember. But in prokaryotes, translation is coupled with transcription. Okay, they are coupled. This coupled nature of transcription to translation actually helps the bacteria to utilize attenuation for the regulation of operon. Without the mechanism of transcription translational coupling, this process is not possible. Why? Let us talk about that in a moment. So at this moment what we need to understand is, let's say the attenuation will come to play when the polymerase, RNA polymerase, can bind to the promoter operator and start to transcribe. Okay, because the tryptophan, even though tryptophan is present, it fails to prevent the polymerase to bind to the promoter operator region. So the presence of tryptophan already failed. It is there, but it already failed. Then only the attenuation will be in place. So at this moment what goes on here is that polymerase binds and what polymerase is doing, they start to transcribe, and as they start transcription, so the transcription is done. So let's imagine this is the mRNA. So from DNA, now we are looking at the structure of mRNA. In this mRNA, what they produce? They produce a leader sequence, and the leader, let's say this is the leader sequence, and rest of them E, D, C, B, A. Now in this leader sequence, we have 4 different individual clusters, okay, 1, 2, 3, 4. 4 different sections in the leader sequence. Now why this is very important is that these sequences are in place in such a way that they have a lot of self-complementary structure. That means 1 can bind to 2, 2 can bind to 3, 3 can bind to 4. Self-interaction of RNA, because RNA is single-stranded, you know, it is single-stranded like this. So single-stranded structures can stick to each other like a velcro. Okay, so the 3 and 4 can form structure like this. 3 and 4 can form structure like this. Okay, 1 and 2 can form structure like this. 2 and 3 can form structure, a little different structure like this, 2 and 3. So they can interact to themselves and they can self-ligate like this. So this is the place of ligation, okay, and the rest are stem structures. So stem loop and all, this sort of structures are quite common. Now imagine one thing, if these are the 2 set of structures that I draw here, right? This is one set of structure where there is a stem and there is a loop. So this stem loop that is formed between 3 and 4 is known as terminator loop structure. But here the structure you can see more like a tree, where we not only have one stem but we have multiple stem and multiple loop in nature. This is known as anti-terminator, okay, anti-terminator loop. So these are the 2 set of loops that can be formed while the mRNA is already transcribed from the DNA. And when this sort of loop structures are formed, you can see from this DNA, RNA, now we are talking at RNA level. But when this loop will form, when they'll get enough time to be folded, because this is a sort of secondary structures of RNA, and the secondary structures will only form when the environment is feasible, and also they get enough time to do so. If some protein cover up this area, then they will not get a chance to self-ligate. Remember, single strand binding proteins protect the DNA single strands from being self-ligated. So similarly, if proteins are associated, they will not allow them to reanneal. But if there is no protein, then they will reanneal. Now the question is, if the tryptophan is already present in the environment, then what happens is that here we have this 1 and 2. So this first sequence is a place where multiple stretch length of nucleotide is there, and 2 codons are out there for tryptophan. 2 codons of tryptophan are present in this mRNA. Okay, so remember the codon. What is a codon? Triplet, 3 nucleotide together, like AUG is a start codon, UGA is a stop codon, and let's say other 3 letter codes are there coding for different amino acids. So 2 set of codons are present there in the sequence number 1. They code for tryptophan amino acid. So now we are combining this transcription to translation. And remember, in this transcription part, although they are transcribing, but the translation is not done yet and the transcription is still not completed. So at this moment, the ribosome start to interact. Okay, and understand this idea now. The ribosome is translating the RNA to protein. And why we call it transcription translational coupled? Because transcription is going on, translation is going on at the same time. The portion of the RNA which is transcribed, ribosome is loaded, translation is going on in that portion. So at this moment, what happens is they will bring tryptophan. So we know the tryptophan is already present in the environment, and when the ribosome binds there, there will be tRNA. If you recall, tRNA will bind to them and they'll try to bring tryptophan, and then they will bring tryptophan quite easily because it's already available in the environment. So the ribosome continue to pass, they will not stop there because the tryptophan is readily available. When they ask for tryptophan, they'll bring tryptophan quite easily, faster, they'll move. So as ribosome continue to move between 1 and 2, so ribosome is a big protein and ribosome structure, which big protein and RNA structure, so which will cover a lot of area between 1 and 2. So 1 and 2 is kind of covered by the ribosome here, and 3 and 4 is now free. So 3 and 4 free. So as the 3 and 4 is kind of free in here and ribosome is covering 1 and 2, this 3 and 4 can self-ligate to form stem loop structure, and when the stem loop structure form between 3 and 4, that is a type of terminator loop. Terminator loop means it is a loop that will terminate the process of transcription. So it will end the process of transcription, due to the formation of terminator loop. So transcription will be ended, no further transcription of the structural genes would be possible. When there is tryptophan present in the environment, then this thing happens. Got it? Now if you don't understand in one go, rewatch this section couple of times and you will understand. Now imagine a situation when there is tryptophan, no tryptophan. If the tryptophan is absent, what will happen if the tryptophan is absent? If there is no tryptophan and the ribosome, the same thing, the leader peptide 1, 2, 4, 3, 4, all the sequences out there, the structural genes are made. Ribosome is also sitting in the sequence number 1 in the leader sequence, and as the tryptophan is not present in the environment, so the ribosome sit there in the sequence number 1, ask for tryptophan, but it failed to bind any tryptophan because there is no tryptophan in the environment, and as there is no tryptophan, ribosome covers up sequence number 1. Ribosome failed to move from 1 to 2, so now ribosome only fills the sequence number 1. So again let me draw here. So we have this and the sequences, sequence 1, 2, 3 and 4, and rest of the structural genes out there. So again remember, ribosome is sitting on sequence 1, that makes the sequence 2 and 3 free. 2 and 3 and 4, all of them are free, but the sequence 2 and 3 are going to self-ligate, self-anneal, not ligate, we say self-anneal, and as a result of self-annealing between 2 and 3, these two leader sequences, they will form this sort of loop known as anti-terminator loop. So the anti-terminator loop is actually formed between the leader sequence 2 and leader sequence 3. So when this anti-terminator loop is formed, this loop means ribosome stops there for a while when the tryptophan is obviously in place. Now it will take some time for them to bring tryptophan. If they bring tryptophan after some time, then the process can continue, but if there is no tryptophan, ribosome releases itself, but the RNA polymerase continue the process of transcription. So the transcription continues. Remember, transcription and translation both are taken place in the same time. Translation is not possible here, but the transcription continues. So the structural genes, that these are E, D, C, B and A, all the structural gene and the products are continue to be made in this second mode where the tryptophan is absent, and this is near to the operon to be turned on. Okay, that is the regulation of attenuation. When the tryptophan is absent, operon is on. Tryptophan is present, operon should be off. Okay, so when the tryptophan is absent, the operon is on. That is quite easy. We don't need attenuation to do that, but attenuation's most important role is to turn off the tryptophan operon when the tryptophan is present in a situation where the polymerase continue to transcribe even though the tryptophan is present. Then attenuator's job is to further prevent the production of tryptophan, because there will be loss of ATP, loss of energy in the cell. This is something that you need to understand. Three important concepts are discussed. One, the transcription translational coupling. Second is terminator anti-terminator loop, and the third is the role of attenuator is maximum in preventing the operon of tryptophan in the presence of tryptophan. not in the absence of tryptophan. If you understand these 3 points, you will never ever forget tryptophan operon in your life. I believe you have a clear idea. If you like this video, please hit the like button, share this video with your friends, subscribe to this channel to get more videos like that in future. And also remember one more thing, if you want to understand lac operon, I have a video on this, you can watch that. Thank you, bye.